Okay, so it starts recording. Okay, guys. Hello one more time. Um, I open the presentation and I will introduce myself just a little bit because I guess it's very important. Um, we will be, I mean, we'll have the lectures and tutorials through this semester, so it's very important to know each other. I'm Duru Mamrikishvili, but everybody calls me Duru. You can call me Duru also. Uh, yeah, I have studied Tbilisi State Medical University, Faculty of Medicine. I also studied as an Erasmus exchange student in UK, University of Chester. I do cardiogenetics training course. Right now, I am studying and working as a doctor in a cardiology hospital, a cardiologist, the Chapitze Heart Center, and I'm working here at the university at New Vision University as a lecturer and facilitator of pathology, pathophysiology, sometimes pharmacology, and PBL. Um, and I used to also teach medical genetics at Tbilisi State Medical University, and I used to work as a junior doctor and doctor in many clinics. But this is just a few sentences about me. Okay, before we start talking about our subject, I want also information from you guys, your background, where are you from, how old are you, um, 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 what do you study actually, so and so. We will start. Okay, Abd Al Latif Kadan. Could you start? Could you tell us
Yeah,
about?
yeah, yeah. So my name is Abed,
Mhm.
from Israel. I'm
From
studying newly nursing. I transferred from a medical doctor, so now I'm in a nursing care program. And
okay,
yeah, I'm 19. Yeah,
okay. Thank you, Abid. Yazan, what about you? I guess Solomon left us. Yazan, could you hear? Uh, let's
Yes, yes, I hear you.
so let
My name,
okay,
yes, my name is Yazan. I am from Israel.
okay,
I'm studying nursing.
mm-hmm. Okay,
Yes, I am 18 years old. 18.
okay, 18. Okay, guys.
Yes, and nice to meet you.
Nice to meet you guys also. Thank
Thank you,
you.
thank you.
Yeah, and I want to tell also something about the situation, what's happening now in whole world. It's a bad situation because of this novel coronavirus, but I hope
Yes,
everything will be okay, and I really believe that we can overcome this. um, with all these we obey all these recommendations from the World Health Organization, our government, and we can uh, yeah, we can overcome this and we can continue studying in our classrooms at universities. And so both of you studied in the nursing program, and in this battle against the coronavirus, actually actually not doctors, but yeah, I think the nurses are in the front line. They are doing lots of things in the clinics. From my experience, and I'm working right now also in the cardiology hospital, and nurses, they are doing lots of things. Yeah, they have more communication and more contact with patients than to doctors. So it's very important profession. And today's we see, yeah. Unfortunately, Solomon left us, but I guess he will join back, and we will yeah, we will ask about him. Yeah. So if you want to tell something, you can yes, say it or message me, and I will ask you some questions also from time to time. Okay, guys, so let's continue my presentation. Actually, I used this presentation also while I was recording the lectures, and all of these materials it will be also in lecture, and you can watch it. So now about our subject, book, and everything. Uh, um, um, so it was a very important aspect. So which book you will gonna to study. But first of all, about subject pathophysiology. Uh, Pathophysiology is sorry. Solomon is back. Solomon, could you hear us? Will you introduce yourself? If you don't want to introduce, it's okay. Could you tell a little bit? Had network issues. Okay. Okay.
Yeah,
Okay.
listening.
Okay, could you introduce yourself? Could you tell us about yourself? Where are you from? How old are you? What are you studying in New University? And I don't know.
Okay. My name is Solomon. I'm from Nigeria. I'm
Okay.
studying physical rehabilitation,
Mhm.
and also I'm 25 years old.
Okay. Okay. Thank you, Salman. Nice to meet you.
Nice to meet you too.
Okay. Yeah. As well. So we will continue now presentation. We was talking about the subject. So pathophysiology is one of the most important subject in basic subjects. Um, because in normal anatomy, histology, physiology, molecular biology, was studying the normal things, how normally body functioning. But in pathophysiology now, we will study all the pathologies, all the disease and pathophysiology of the disease. It's very crucial for understanding the mechanism, and it's very important. And I like when the doctors, nurses, rehabilitation people could think and guess the disease. I don't know, and I think the only way. Only way to get the skill to guess the disease and understand the topic is to know the pathophysiology, to know the mechanism. So pathophysiology is a very important subject, but because you are studying nursing and medical rehabilitation, you need a specific way, you need specific information because you should not study the same book as the biology faculty students studying, or the medical students or stomatology students, and you need different book because yeah, because you yeah. So I choose this book, The Fundamentals of Applied Pathophysiology. I guess this book must be uploaded in your portal because I sent them this book. If you don't have the book, you can ask them, and also I also have the own free PDF version, so I can also send to you, because till that time, for this time, we will use this book as a main book. So third edition, yeah, this is a British book, and I really liked it. Okay, so what will be objectives of our whole course? So all this is overarching objectives. The overarching objectives will be, yeah, what we already mentioned, to understand the pathophysiology of the disease, understand the mechanism. We will discuss all the organ systems and all the pathologies of the organ systems. Here are the content of the book. So first of all, we will start, and I will remind you, because I guess you are supposed to know from molecular biology, I don't know, physiology, anatomy, you should know about cell tissues, but I will remind you because it's very crucial. You should know, you should have the background. to understand the pathophysiology. We will speak about cancer, inflammation, shock. Then we will continue and discuss different organ systems, for example, nervous system, heart, vascular, blood, renal, so and so and so. And this is very interesting for me also. Guys, so I'm teaching almost 3 years. I have 3 year practice of teaching. I actively use during my classes, during my tutorial, active learning and adaptive teaching techniques. But I don't know, but I'm using those techniques during my classes when me and my students are sitting in the classrooms and we have the face-to-face communication, but I don't know how we can do it now in online. But let's see. We can manage somehow this. So this is all the techniques I'm using. Yeah, most of them at least, but many of them need some face-to-face interaction. But let's see, has 3-2-1 technique, and I have planned to use this technique today. One of my favorite technique is a jigsaw reading, but I guess we can't do it online. Yeah, another facilitating tool I'm using quite often. Because I was teaching also problem-based learning, PBL, and pathology and pharmacology. This is a Lego Serious game. Have you ever heard about Lego Serious game? You know, of course, about Lego, right? Those things when the children, when the children are playing with this. But what about Lego Serious work and game? Have you ever heard about it?
Actually, this is my first time hearing about it. So, no.
Okay, yeah, thank you. Yeah, it's quite a new thing in the world of the facilitation and teaching. So what does it mean? I don't think we can do it online also. I don't know. You can manage if you have the Legos at home, but it's okay. We can speak about it later today or in other lectures. But I will tell you the concept of the Lego Serious game. The Lego Serious game is the concept that by the help of the Legos bricks, uh, to understand the concept, meaning of the different topics. So this facilitation tool is used in many conferencing, different workshops, different classes. So what they are doing, first of all, you should know the rules, how it works. So as a rule, in first classes, I was introducing my students the rules of the Lego Serious games, how to use it, and they are there we have the exercises to get the skills, and then incorporation of the Lego Serious game in our actual studying. And I was I'm asking and giving them a task, for example. Builds a model which represents I don't know, cell membrane, which represents cell, cytoplasm, connective tissue, and explain this, explain the cell concept of the cell by the help of their model. So this type of facilitation tools helps them to understand the concept, externalize all the meaning of the concept, all the meaning of the topics, and it was really good. But okay, let's see. Maybe we can manage it somehow if you have the Lego bricks at home, but not today, I guess. This is the example from this book. Okay. Okay, guys. Now let's start our very first chapter. This chapter will be about cell and body tissue physiology. So as I told you, this is just reminding for you because you are already supposed to know this. Till now, me and your coordinator Salome, we spoke to each other, and we have this plan that I will record approximately 1 hour lecture, and you will watch this lecture. As I told you, I already record a lecture for these topics, and then during online classes, we will have the interaction. We will discuss more deeply. I will ask some questions. I'll give you some activities to do, and we will have the active learnings together. So, Solomon, what about you? Did you watch the videos I record lectures? Is Solomon here?
Yes, yes.
What?
Actually, not yet.
Not
Actually,
yet.
not yet.
Not yet.
Yes.
Okay. Okay. It's okay. Um, yeah. So I guess my this very first lecture, this very first online class will be like more. I will speak more, but I will ask you sometimes some questions, and because I don't want to speak monotony, and it's not interesting. Yeah. Okay.
Okay.
Yeah, yeah. So we will speak about cell and body tissue physiology. So it's recording. Sorry, cell and body tissue physiology. Okay, here are the objectives of today's lecture and tutorial. So what we are gonna to um, speak about. It will be about anatomy of the cell membrane, cytoplasm, nucleus, mitosis and meiosis, organelles, type of cells, different types of tissue and tissue repair. Uh, um, Guys, also, it's very important. That's why I like face-to-face communication and the classes, because I always encourage myself, first of all, that um, I should understand what you need. What you need from this lecture, what knowledge, what background you have already. It doesn't matter if you know good, very well, pathology or physiology or anatomy, it doesn't matter. But I should know about it, because according to this information, I can conduct all classes. And according to this information, we can make our tutorial and lecture more productive, more beneficial for you guys. So first lecture will be about cell and tissues. Um, so what do you know, guys, already about human body cells and different tissues? Uh, I guess you already studied several subjects, right? Did you study physiology, anatomy, biology? Because I don't know this your nursing and rehabilitation faculty program. Could you tell me a little bit about your subjects you study?
In the first semester, I studied life science
Okay, so what?
and anatomy, body system.
Okay, okay. So
Uh,
hmm,
yes.
this is actually second semester or third semester?
Now this is the second semester.
Second semester.
Yes.
Okay, okay. Thank you. What about others?
What?
What about other students?
Oh,
It's actually my second semester. I learned my first semester during the medical doctor program. So I
Okay.
learned about body system and life
Okay.
sciences. Yeah.
Mm-hmm. Okay, thank you. Thank you. Okay, and what about third one? Okay. So you know about cell and you know about the tissues. What do you know about cell? Which parts are found in the cell? Could you tell me, one of you?
I mean, there is two major types: the eukaryotic and prokaryotic cells. Yeah.
Exactly, exactly.
Yeah, inside the cell we can find the organelles, of course, and surrounding of course, the organelles are present, but and
Mm-hmm.
yeah, and eukaryotic cells they are like enveloped in like a membrane.
Mm-hmm.
There is a double membrane happening, layers happening in eukaryotics, unlike prokaryotic cells. So yeah.
So what's the difference between eukaryotic and prokaryotic? Is it about membrane or
It's about mainly it's about the DNA situation. So and uh,
nucleus, right?
yeah, yeah. So yeah, and eukaryotic cells they are like enveloped inside a membrane, specific membrane, and uh, and and prokaryotic cells they are just flowing in the cytoplasm. So
But I guess it's vice versa. Prokaryotic cells have the nucleus, so their DNA is enveloped as
Yeah,
an
it's like the DNA inside the nucleus in
in which
the
cells?
yes, but in the eukaryotic cells in cytoplasm the DNA.
Yeah, yeah. So
Yes.
Abd was so he mentioned all these main topics. Yeah. Thank you, guys. Thank you. And there's different organelles. So, which organelle do you know? The main organelles.
maybe the mitochondria, the Golgi apparatus,
Exactly.
the rough and the smooth endoplasmic reticulum, the nucleus,
Exactly.
of course. Yeah.
Mhm.
Uh,
Well,
what else? What else? Mm,
yeah, I guess you mentioned almost all of them. Ribosomes. Yeah. And we will discuss all of them also to you today. Okay, guys, what about tissues? What what about tissues? What type of tissues do you know?
I mean, we have connective tissues, then we have types and the epithelium tissue. So
Yeah.
that's five major types. Yeah.
So connective tissue, epithelium tissue. Uh, okay, thank you. And we have two more tissues, right? This is nervous tissue, epithelium tissue, connective tissue, nervous tissue, and what about first one? First one is oh my god, muscle tissue. Sorry, muscle tissue. Yeah. And we will discuss all these tissues, and I will tell you the exact examples of the all these tissues. Okay, guys, thank you very much. I really appreciate your um, interaction, because I guess it's the best way to have the tutorial. Um, okay. So this very first lecture will be according your book, um, because yeah, but in other lectures I will bring also slides from other topics. But I really liked your book somehow. It's easy to understand, to read also, and yeah, it's comfortable. So there are the keywords. So plasma membrane, organelles, connective tissue, passive transport nucleus. Yeah, all these words will be mentioned, keywords. Okay, here is test your prior knowledge. So what do you know till today? Actually, we already did this, but just go quickly through it. So what are the three main parts of human cell? Uh, okay, guys. Well, what are the three main parts of human cell? That's not. We already mentioned all of them.
So maybe the nucleus and the genetic materials, and then the organelles and the cytoplasm and the membrane last. Yeah.
I guess yes. I guess you are true. Yeah. So membrane, cytoplasm, and nucleus. Yeah.
Uh-huh.
Thank you. Thank you. So describe the structure and function of a human cell. Okay. What are the functions of human cells. So why do we have cells? Why do we need them?
I mean, for many stuff, it depends on the type of the cell and the
Yeah.
place. Yeah.
So what are the so tell me one example and
Like
function.
maybe reproduction functionally, and yeah.
Yeah, yeah,
Uh-huh.
yeah.
Uh-huh.
Uh,
Transport the blood.
trust.
Growing,
Mhm.
growth.
Yes.
Yeah. Yeah, and it was actually true when you mentioned that it depends on the type of cell and type of tissue, because every cell has its own function. Yeah, so yeah, blood cells have the opportunity to transport different metabolic products, also oxygen and CO2. We know that neurons, which are the cells of the nervous system, have the function to conduct the electrical impulses. I don't know, epithelial tissue, which covers the skin, it has a function to cover and protect. Lots of functions. Yeah, yeah. We will may discuss many of them today. Okay, now about phases of cell cycle. What do you know, guys, about cell cycle?
I remember it as MAT, so BMAT. Yeah, so the prophase,
Okay,
MAT.
yeah, yeah,
Yeah, yeah.
yeah,
Yeah, so there is meiosis and mitosis.
yeah,
Uh,
yeah,
yeah, one's happens for the gametes, for the
yeah.
yeah, and one's for normal cells, like yeah.
Exactly. Which one? Which one for which? Mitosis and
So
meiosis.
that's the hard one because I always make mistakes. Yeah,
Yeah,
um,
it's
yeah.
okay.
So I think mitosis is for normal cells, and meiosis is for gametes. Yeah.
Yeah, you are true. Yeah, you are true.
Mhm.
That's correct. Okay, thank you, guys. So, what about cycle? Because cell cycle, everything and everything in the life, in this universe, in this world has a cycle. We also humans have a cycle. We are born, we I don't know, raised, we have the families, children, then go old and dead. That's a normal thing. That's how it works. And cells have also the cycle, but I don't think it's a proper. I mean, yeah, I was talking about not life cycle for cell, but the different cycle. These cycles approximately takes 24 hours, and each contains interphase, which is approximately 23 hours, and only 1 hour is mitosis. Yeah, which in normal cells, in almost every cells. So during mitosis, cell is divided. Then there's prophase, metaphase, anaphase, and telophase, and we will discuss all these phases. And what's happening in interphases? During interphases, DNA is replicated, all the organelles are divided, energy is provided, and so and so, and that's continues and continues. How, yeah. But I will mention also here there are some cells which cell cycle is different, because they don't divide that often. Maybe it's
The
not
G zero phase,
zero phase exactly.
like neurons, I think.
Yeah, exactly, man. Yeah, so neurons are not dividing, therefore they are in G0 phase, and how they live, they are in their own, like, not their own. They are they have lots of impact of our lives. Neurons. Yeah. Okay, guys. Yeah. So let's continue. This was about a cell cycle. Make a list of the major cellular organelles. Uh, yeah, we already mentioned this. So mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and so on and so. We will speak. So name the 4 tissue types and explain the differences between them. We already mentioned that. Connective tissue, epithelial tissue, muscle tissue, and nervous tissue. Yeah. Okay, could you tell me the function of each tissue? So what's the function of, we mentioned somehow a function of epithelial tissue, but could you tell me the functions of each tissue? Or what's the function of them? Somehow we mentioned also all of this.
So I think it's like close to the name of the tissue, so we can
Mhm.
know from the name. Connective tissue, it connects like things together. Nerves, we can know about nerves that they are like transmitting like
Yeah.
yeah, so electrical signals,
Yeah.
and we can know about muscle tissues that they are supposed to function as like
Right here.
flexing, extending, and epithelium. I think I forgot actually, but I think it's about like protecting the body. So yeah,
Yeah, it's
yeah,
covers. The skin is.
like covering, yeah, protecting, yeah.
Yeah.
Mhm.
Okay. Thank you, man. Thank you.
Mhm.
Thank you very much. Okay. Okay, guys. By the way, where are you guys right now? Are you in Georgia or back to Nigeria and Israel? Where are
I meant
you?
I'm still here in Georgia, actually.
You
Yeah,
are
I'm in
still.
Georgia too.
Okay. Okay. So you decided to stay here, or what's the case? What's the case?
I've always been in Georgia for the past 5 years now. So
Okay. Yes, Solomon. Yazan message does in which alphabet is it?
It's we can spell it as Nor.
Huh?
Nor.
Nor.
Yeah, I'm still here. Yeah,
This
I'm still
lecture,
here in Georgia.
yeah, it's fun.
So I
Ah,
mean, the lecture is good. It's
Yeah,
perfect.
yeah.
okay. And yeah, I hope you are safe and oh, yeah.
Yeah, thank you,
Yeah.
thank you.
Okay. Okay, guys. Because it's quite a difficult situation, but I guess in our country, in Georgia, it's more or less normal situation to compare other countries. But we does not know what will happen in future. I hope everything will be okay. Okay, so let's continue. So test your prior knowledge. So learning outcomes, guys. We on completion of this chapter, the reader will be able. What will you get from this chapter? So outline the structure and function of the human cell. List and describe the function of the organelles, explain the phases of the cell cycle. So you will be able to do this, explain the cellular transport. Actually, you already are able, you know all this information, but I will just remind you because it's very first tutorial and this information is very important because I always think without fundamentals, without prior knowledge, without normal physiology and anatomy, you can understand pathology, of course. It's describe the structure of function, epithelial connective, explain the process of tissue repair, inflammation. Okay. Okay, so we can start the introductions. So, from current slide, I guess it will be better. So, so all cells have many characteristics, but these characteristics can differ from cell to cell, such as cells are able to carry out specific functions. They are active, right? Yeah, I like this second sentence. Cells need to consume food to live and to carry out their functions. Also, they don't have mouths. They are still able to catch and digest their food and use it for growth and reproduction. Of course, cells need also food, but yeah, no, it's not like we eat, yeah, they're using different amino acids, different, I don't know, carbohydrates. Yeah, the correct term for this endocytosis, and by the endocytosis, they are taking this food, they surround and engulf organisms such as bacteria, and digest them. Cell can grow and repair, of course. Cells can reproduce themselves by the fusion, and we will speak lots about today about this fusion. But I guess I have to hurry up because we don't have that much time. Though, like human cells, can be irritable. Yeah, they look like us if something upsets or stimulates them. By the way, guys, I guess you prefer that we should have the break because we are supposed to have a 2 hour lecture, right? So we are supposed to finish at 7 o'clock. Do you need break between one lecture, between these two lecture?
I
Like,
think
yeah,
at 6 o'clock we take a 5 minutes break, and
I
that's it.
agree. I
Yeah,
agree. I
yeah. I guess that's so we'll have break and then continue. Okay, guys. So let's continue. So the nutrition that cells take in is also used to for the storage and release of energy. Yeah, these nutrients are the source of energy, and this energy producing process takes place in one organelle. Do you remember the organelle where the energy producing process takes place? Hmm?
I think the energy is produced via the mitochondria. So yeah.
Exactly, man. Yeah.
Mhm.
Yeah, there is a TCA cycle, tricarboxylate cycle, tricarboxylate, yeah, or not TCA cycle, and this is a main source for energy for prokaryotic cells, but in eukaryotic cells, main source of energy is glycolysis, a breakdown of glucose. Okay, so similarly, just as humans don't utilize all the food they eat, some of the it cannot be used, and so is excreted. Cells excrete what they don't need or cannot use. Yeah, just all humans will eventually die, so will cells. That's how it works. Yeah. Some have a short life, whilst others survive many years, but eventually they will die. Sad story. So cells are not all that different from humans in many aspects. They are the part of us, and they do what humans do, but in other ways. Yeah. Okay, guys, we mentioned we already spoke about the anatomy of the cell, but here's the picture, and I will show you quickly. So uh, this is a cell membrane. This is microfilaments, which are not found in all cells, but some of cells have it. For example, it's quite commonly found in where is it found? Do you remember, guys? I'm not saying that you are supposed to know this, but maybe one of you know which cells in which organ do microfilaments have.
Organ. So I think the cell that have it is a cell that moves.
Yeah.
So maybe the egg cell, maybe. Ha-
Yeah, and spermatocytes, yeah,
Mhm, mhm, yeah.
uh, yeah, yeah. But that's, I guess, flagellum. Yeah, thank you, man. Yeah, but microfilaments, it's a movement. Yeah, you are true. As far as I know, it's also found in the respiratory tract.
System. Yeah. So we cough. That's why we
Yeah,
cough.
yeah.
So, yeah.
And yeah, also it's found in GI tract, gastrointestinal tract, because these microfilaments, you see here, also increase surface, right? Increase the surface of the cells. They are
So in the intestines, I think there's a
sorry?
lot.
Yeah,
In the intestines, there's a
yeah,
lot.
yeah, exactly, for reabsorption, different breakdown products. Yeah, yeah. Thank you, guys. So microfilaments, flagellum, lysosome, the organ in the as a, so what's the main function of the lysosome? What substances are found inside of the lysosome?
Lysosome, it's
Okay.
for cleaning the unfunction
For the waste, yeah.
organelles. Yes.
Exactly. And what substances are found inside of lysosomes? Uh,
Again, can you repeat the question, please?
which substances, which substances are found? What's a
A lot
gen?
of enzymes, I
The
guess.
enzymes, yeah, different enzymes. Yeah. Okay, thank you, guys. Thank you. Different enzymes, and by the help of these enzymes, different nutrients are break down. Yeah, even some bacteria or viruses which enter the cells or are endocytosis and engulfed by the cells, they are break down in the lysosome or phagolysosomes. Okay, we will mention of lysosomes also later today. Okay, here is a endoplasmic reticulum. We have smooth endoplasmic reticulum and rough endoplasmic reticulum. You see here. So, what's the difference between them? And yeah,
So the rough one have a lot of ribosomes on it. That's why it's like a rough appearance looks,
mhm,
yeah. And
mhm.
yeah, they prepare like uh, s- um, some
Yeah.
molecules inside. Yeah, yeah. So basically, it prepares some proteins and stuff. Yeah.
Okay, thank you, ma'am. What about the function? What's the function of the endoplasmic reticulum? Why do we have in the cells? Why the cells have it? Mm. The function, for example, rapid endoplasmic reticulum, as you already mentioned. Yeah, okay, Yazan, did you say something?
Uh, I think for transport the protein to the
Yeah,
nucleus.
So yeah, in sacks, I guess, in like
Transport.
really small envelopes, in sacks, like Latron. Yeah.
sucking, yeah, or what does it? No, yeah, yeah, and the ribosomes are found on the surface of the rough endoplasmic reticulum, right? So, what are the ribosomes? Ribosomes
So
play
mainly for making proteins.
breaking proteins, synthesizing proteins. So proteins are synthesized in this on the rough endoplasmic reticulum. Different products are transported and stored. Different carbohydrates, different reactions takes place, and so and so. Yeah, yeah, guys. Thank you, thank you. Um, really, I really see that you have the knowledge, and that's really good. Good. That will help us to understand the pathology. So microfilaments, microfilaments which are found on the, which are quite close to the membrane. Yeah, and they are different microfilaments. They play important role to make the shape of the cell. For example, when you are talking about one type of anemia, which is spherocytosis. Actually, what does anemia means, guys? Mm, do you know
It's
what?
like
Mhm.
it's like I think there's like two types of anemias. There is a quality problem, and there is a quantitative problem. So with like how much there is red blood cells.
Yeah,
Yeah, and I think the cell cycle is faster, so the cells die faster, and
exactly. Yeah.
and some time of anemia. So yeah, there's two types, I think, the S and the C. I remember.
This is when we are to sickle cell anemia and the hemoglobin C, NS, right?
Yeah, yeah, and there is one which is like CS, like together.
Yeah, exactly. Yeah.
there's a problem with the both genes. So yeah.
Yeah, and they are the heterozygotes for both of these disorders.
Yeah, exactly.
Yes. Okay, man. Thank you. Yeah. So anemia is about red blood cell disorder. So and yeah, there are different classification of anemia according to this volume of the red blood cells, so microcytic, normocytic, macrocytic. We will discuss these topics when we'll speak about red blood cell disorders. But why I mention here, because one subtype of anemia is anemia, spherocytic anemia, when the red blood cells change their shape and they became like sphere and
Like
spherocyte.
not like the moon. Like there is like a type of like it's like a moon in half. You know.
Yeah.
Yeah.
But when you are talking this, you are talking now sickle cell anemia. This is
Yeah.
yeah,
Sickle cell.
yeah. But I'm talking about spherocytic anemia. This is different.
Yeah, this is new.
Yeah. Why I mentioned this? Because to development of this anemia and to change the shape of red blood cells play important role microfilaments. For example, ankyrin and Benford 2.2. These microfilaments, because of the damage and genetic problem of those microfilaments, shape of the red blood cells is changed and they became spherocytic, and because of they became spherocytic, they can be occluded and in the spleen especially, and can be hemolyzed and can cause anemia. So that was the main thing why I mentioned this. Yeah, so microfilaments have the important role. Thank you guys one more time. So Golgi complex, Golgi apparatus. What about Golgi complex? What do you know about it? What's a function of the Golgi?
I mean, I remember from the shape that
Yeah,
that Golgi complex like prepare like some molecules in like sacs, which is like an envelope
yeah,
that make like substances travel throughout the cell and out
yeah.
maybe. Yeah.
Exactly, guys. Yeah, and yes, lots of substances which are produced in the cell. No, yeah, during their production, and yeah, they ended up in the Golgi apparatus, and they, for example, different hormones which are produced. Yeah,
Like,
final steps.
yeah, I think mainly proteins and fats.
Yeah,
So
yeah, exactly, man. Yeah, we will mention also Golgi apparatus. About ribosomes. Ribosomes. There are many types of different types of ribosomes. Not that much. One of the important function is take a part in the protein synthesis. Yeah,
making RNA, I think.
making what?
RNA, the RNA ribosome.
Yeah, there's RNA ribosome also. Yeah, yeah, ribosomal RNA. Yeah,
Yeah,
which
I think it's more in the nucleus, but it's not in the
yeah,
cytoplasm. Yeah.
yeah, exactly, man. About cytoplasm. Cytoplasm is a space which is mainly filled with fluid like liquid, and approximately 75 to 90% of this fluid is the water, and all these organelles are in the cytoplasm. And now what about nucleus, guys? What about nucleus? What's the function of nucleus? What are they found inside of the nucleus?
So I think the main idea of it is to protect the genetic materials and
Exactly.
to like make it like move from place to place, and so get like stuff in and out from the nucleus. So it's like a barrier.
Exactly, guys. It's a barrier. It has double layer membrane, and inside of the nucleus is chromosomes, DNA, yeah, right? Genetic information, and we will discuss this information. And last one, okay, here's the cell membrane. Okay, I will tell you myself about cell membrane, and I'm sure you know about cell membrane. Cell membrane, which covers the cells. Cell membrane has a lipid bilayer, so bilayer of the lipid. Each layer of a lipid contains lipids which have the polar head and polar head, which means that it's hydrophilic. What does hydrophilic means?
So they can interact with water.
Yeah, they love water, right? Yeah, hydrophilic, exactly. And inside, so tails are fatty acids. They are hydrophobic, though they have the phobia of hydrophobia, so they don't like water. Yeah. Uh, that's why actually this is a clever idea. Of course, the hydrophilic parts of the bilayer are outside because they will have the contact and communication to the extracellular and intracellular matrix, intracellular space, and these spaces mainly contain water. Yeah. Yeah, cells are clever and wise. They know how to do things, how to manage things. But hydrophobic ones are inside of these hydrophilic heads. So this is the main part of the membrane, but other part of the membranes are the integral proteins, transmembrane proteins. So proteins which are found inside of this cell membrane, transmembraning, and there are different types of proteins that can be the channel protein, for example, this one. This is channel protein. What does it mean? What does channel protein means?
So I think it transfers products passively, so without energy. So specific molecules can like just go through it. Yeah.
Yeah, ions. Yeah, for example, potassium, sodium, calcium. It depends on, yeah, exactly. Yeah, and it can be passive and it can be active also. Sometimes we will discuss this. They are peripheral protein. They are also glycoprotein, glycolipid, carbohydrate. Okay, sorry, glycoproteins. Uh, so this is the glycoproteins, carbohydrates, or this violet one is carbohydrate. Cholesterol is also sometimes here. Yeah. I guess, guys, now we all deserve to have the break, and it's essential to have the break. Let's have how many minutes breaks? Let's meet each other at 6. Let's have a 10 minutes breaks. Let's meet 6 to 6:10. Um, but I don't know. Should we just mute our conversation or turn off it and then turn on? But what do you think?
I mean, if you want to still record, you have
Yeah.
to keep on the convo like open.
Oh,
Uh, but you can like start a new record like after the break. We can leave and start a new record.
it's okay for me. I can have it open. Just I will mute it, and we will meet in 10 minutes. It's 6:10. Yeah.
Okay,
Stop.
that works.
Okay. 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 What about Diazan and Solomon?
Yeah, no problem.
No problem. Okay, guys. Okay, so I will mute. Mhm. We will continue. Uh, Okay, my students, I'm back to the lecture. Are you also here?
The Zoe is substantially
Yes,
more
I'm here.
expensive to buy than conventionally powered rivals like the Ford Fiesta.
Okay, so you're here. Okay, guys. Why is it? Why am I into? What is the presentation? Okay. So before we continue, so you are here. You know that there are several rules conducted by our government. So what are you doing at home? How you spend your time during this isolation and period? How many subjects do you have at universities? And I don't know how you spend your time. Can you tell me?
For now, I have 2 subjects, and this pathophysiology and the writing and reporting for success.
How is it? What can you repeat? Right.
Writing and reporting for success.
Wow, interesting.
Yes.
Yeah. Same situation or same case with me. I have 2 subjects.
Yes.
What about what else do you do? Are you doing in isolation or
Eating, sleeping. That's it.
A lot of video games also. So yeah.
yeah,
Yes, playing games.
yeah, yeah. I was in previous semesters when I had the students in class. I was asking about their hobbies, what they are doing free time, and many students was telling to me, my hobby and during free time I'm sleeping. I guess it's an interesting hobby, but nowadays everything is changed. Yeah, somehow we should make it because our mental health and mental health is very important also. Of course, corona affects our bodies, and it's very dangerous virus as we see. But during the isolation and during the staying at home whole times, you should take care of mental health and yeah, and should be occupied most of the time. Okay, continue. Let's continue. You see the slide, right? Yeah. So here are the functions of the cell membrane. So functions endocytosis and exocytosis. So what's endocytosis and exocytosis? It's a transport of fluids and other matter into and out of the cell. Endocytosis is the into the cell, exocytosis out of the cell. There are 3 types of endocytosis. So transport of fluid or other matter into the cell. This is phagocytosis, pinocytosis, receptor-mediated endocytosis. What do you know about phagocytosis, guys? Because this is very, very important.
Phagocytosis.
Yeah.
It's done by the white blood cells, so it consumes like any pathogen or a strange strange something from the environment. So without the receptors, so the body doesn't recognize it. So yeah, it yeah.
Yeah, you are true. Yeah. How should I call it? Your name is Abital or
Yeah, you just can say Abed.
Abed?
Yeah.
Abed, form of even in the Bible.
Yeah.
One by Abed. Okay. Thank you, Abed. Yeah. Okay, yeah. So phagocytosis you mentioned quite well, but when it comes to pinocytosis, it involves the ingestion of small particles and fluids. But mainly, it's during pinocyte. When we are talking about pinocytosis, we are talking consuming of the fats. Yeah, and receptor-mediated endocytosis. Actually, I have the figure here, endocytosis. And this is receptor mediated because you see here on the surface of the membrane is the really receptor. This particle connects to it, then it's invaginated plasma membrane, and vesicle is formed. Outside of this vesicle is a clathrin coated, clathrin, it's called clathrin coated. Then uncoating takes place. Then we have uncoated vesicle. This vesicle then fuses, make fusions to the endosomes. This is transport vesicle, and this transport vesicles transports these receptors. Yes, and the recycling of receptors to plasma membrane, yeah. And all these products, all these substances is ended up here. Then here you will see how this transport vesicle make fusion with the lysosome, and these substances could be break down in the lysosomes. We already mentioned in lysosomes there are many enzymes, and they make breakdown, and all these little particles can be used for the cell, for a different perspective, as the use of the building material or for the energy, and so on and so. Okay, transport across the cell membrane. This is very important also. So we mentioned there are the transmembrane proteins, and those proteins very often play the role to transport different ions. Also, some particles can go through the membrane and be transported inside or outside of the cell. So the controlled release. Okay, but a very important aspect, very important factors which play important role, which particles and which way they will be transported, is that, for example, is it size of the particle, because some bigger particle could not go through the cell membrane. It depends on the size. It depends on the solubility in lipids, because, as we already mentioned, the tails of this bilipid layer is for hydrophobic. So here only the particles who are soluble in lipids can go through this bilipid layer. Yeah. Another factor is if an ion has an electrical charge opposite to that membrane, because you know that different opposite charges attracts each other. So these ions could be attracted to the membrane and can more easily pass through it. And also, as we already mentioned, carrier integral proteins can carry substances across a membrane regardless of their size, ability to dissolve in lipids, or membrane electrical charge, because those are the transmembrane proteins. And here all these rules changes. Okay, here we have two way of the substance transport: passive and or active. We mentioned this today. Okay, guys, who can speak about passive or active transport? What's the differences between them? And here are the examples. Who know the differences between them?
So we can say the first difference is with energy. The passive does not consume energy,
Okay.
uh, and the active does.
Yeah.
Also, I think the passive is like with the concentration of the ingredients or the
Mhm.
units.
Yeah.
Uh, yeah, it goes like with it, and the active
Mhm.
one is like against it. It is like, yeah. So that's why we need energy, like to push against the flow. Yeah.
Exactly. So we needed an energy to go yeah, against the concentration gradient or the man. You are true, man. And examples. Here we have the examples. So passive process are diffusion, facilitated diffusion, osmosis, filtration. For example, filtration takes place in the glomerulus in the kidney, where the urine is produced. Filtration takes place.
Okay.
Um,
There's the Bauman cup, something like that.
yeah, Bowman capsule.
Yeah,
Yeah,
Bauman cup. Yeah.
yeah, this is very important. Thank you. Yeah. And active processes are active transport pumps. When these pumps actively transport endocytosis, we already mentioned endocytosis, and exocytosis. They are also active. 1 minute, I will unplug my charger because it's unplugged. Okay, here we go. Yeah. Okay, guys. Now, what about diffusion and facilitated diffusion? What do you know about it? Do you know about?
I don't know what's the difference, honestly. So,
Yeah. Well, what does diffusion means?
in English, let me think. Mm, it's like about the flow. Also, I need to say it again. Like it's like a subunit
Yeah.
going through the lower
Yeah.
concentration to like balance the
Yeah.
concentration. Yeah, yeah,
Exactly.
yeah.
Exactly. Substances are going from the higher concentration to the low concentration to make the balance.
Balance.
Because
Yeah.
I believe that in this world, everything and everybody are prone to find the balance.
Mhm.
Yeah. Okay, yeah, that's true. And facilitated diffusion, it's for example, has a good example. Glucose transport. Glucose transport does not sometimes and very often glucose transport doesn't need energy. It's passive transport, but the transport of glucose is facilitated by another substance transport. For example, it's facilitated by sodium transport. If the sodium is transported, glucose will be also, or potassium. So it's facilitated by another substances. Yeah, because I guess they have the same um, pores or same proteins which passively transport them. What about osmosis? What do you know about osmosis?
For water, the
Yeah,
diffusion water, we'll call it osmosis.
that's for water. Exactly. Yeah. And here I have a good example, and I will explain to you osmosis. So this is the first picture, and then will be another picture. So what's happening here? Here we have this container which is divided into part by this semi-permeable membrane. What does that mean? This is semi-permeable. This is permeable, but not permeable for everything. It only allows water, but it does not allow this solute. So, and you see what's happening in the first at first. In this compartment, in this part, there is dilute solution because there is less amount of solute in water, and to compare another one, there is concentrated solute. So lots of solute is found in the water. I mean, and water concentration, water amount is almost similar. I don't know if we can say, but yeah, the dilute solution and concentrated solution. So what's happening then? Because this is semi-permeable membrane, and also water wants to make balance, water starts to be transported from this part to this part, and that will be final result. So water from this compartment starts to transport here, and you'll see equal concentration of solute on either side of the semi-membrane membrane. And this process and this force is called osmotic pressure, and osmosis did take place here. Okay, does it make sense, guys? Do you understand? I don't know if I...
Yeah, it does. So the water rushes to balance the concentration.
Yeah. Okay, even water wants to balance. Okay, yeah. We mentioned filtration. I guess I have to hurry up because there are lots of things and we only left. There's active process, so there are different active transport pumps, endocytosis, as we mentioned, but I had here planned to have the active learning activity, which is called 3, 2, 1. Okay, let's have it. 3, 2, 1. It will take just several minutes. What about other? What about Solomon and Yazan? Are you here, guys? And
Yes, I am here.
Okay, man. Okay. So what we are doing, this technique 3, 2, 1. This is graphic organizers that learners use to record ideas, observations, and the reflections related to a specific learning experiences. Yeah, this is very easy. So instructor provides each learner with the 3, 2, 1 graphic organizer, including prompts, 3 important points, 2 limitations. Okay, this is very easy. In this case, today, how I use how I use. So you have to search and find information and write down this information in this way. 3 important points about active transport pumps. 2 examples of active transport pump, because there are many different type of pumps for different anions or different substances, and 1 question you still have about this topic. Guys, do you understand what you have to do?
So I think we mentioned very important points about the active.
Sorry, Abed, or I don't know who you are. You are okay, guys. This is active learning. So first of all, you have to search by yourself and write down it. And I'll give you several minutes. Let's have I don't know, 4 minutes, maximum 5 minutes. Search information about it, and then I will ask you. Okay? Search information through this way. So 3 important points about active transport pumps, 2 examples of active transport pump, 1 question you still have. Does it make sense? You understand?
Okay, let's do it.
Okay, let's do it just quickly.
Yes.
Okay, write down and we'll continue. Just tell me, make me informed when you will be done. We can start.
So I think I'm ready.
Okay. What about Yazan, sorry. Let's wait for others for a few minutes and then we'll start. Is Solomon with us? Hey man. Hey. Okay, so we are waiting for Yazan. Actually, we can start a bit, and Yazan will come and join. So, what did you search? What did you find about active transport pump? So you have to write down 3 important points. For example, one question.
I mean, first point, they use energy, unlike passive transport. Of course, we say that. Second point, that they sometimes or most of the times they use like assistant enzymes. So
What does
the
it mean?
so there is specific enzymes that they are associated with the function of the active transport.
Yeah,
Uh, yeah.
okay.
And the third point, actually, I learned that there's two types. There's the direct and the not direct transport. Yeah, so indirect one. Yeah,
Okay.
one of those use metabolic energy, and the other like combine molecules.
Mm-hmm.
Two
Yeah,
examples. There is the in like active potential and active membrane potentials. I think there's the the membrane potential. There is the sodium potassium bump. I think
yeah, two.
so. Yeah,
Yeah,
so they push potassium and yeah. And I have a question about like when I read about the indirect one, it says that. They enter like by coupling or combining the molecule.
yeah.
Uh, yeah, my question is actually, does that require still energy, or it just like they just say like the active transport requires energy because there's another like molecule that needs to be there to like couple with a specific molecule that we need to enter into the cell or out.
Mhm. Okay, Abed. So make sure I understand your question. So indirect transport, as you already mentioned, that means that substance, in this case, you use the transport pumps, which is used already for other substance to be transported, right? Am I true or
Yeah,
what's
yeah. There's the indirect,
yeah?
indirect one. It involves coupling the molecule with another moving along.
Yeah, yeah,
Yeah.
yeah,
So that's my question.
yeah. And your question was, does it need extra energy?
Energy still needs energy, yeah.
Yeah.
Like
Uh,
the direct one, yeah.
Yeah, I guess, and as far as I'm concerned, they don't need energy anymore. They are using already this energy. They're already using this energy. But you know, no. But you know, man, I think the correct answer will be that case that these indirect transport pumps, they are existing only in this case. They don't exist, and we can't talk about them separately. So the amount of energy which is consumed for transport during indirect transport for one substance during coupling, this is consumed for both of them, I guess, and we can't speak about them separately. This is how is it done normal way, and yeah, so we can't say yeah, because um, yeah, that's what I think. Do you understand what I mean?
Yeah, I do because there is like I read
Yeah,
one time about like a protein structure that
yeah,
via coupling molecules they can like react in a different way, like maybe change the structure of the cell, maybe make the cell
uh,
move. I don't think they are considered as transporters, but
yeah.
maybe.
They don't consider as a transporters. Ah, okay. So indirect trans. Ah, okay. So maybe I don't understand the topic and the question. Yeah. Okay, man. To tell the truth, I don't know that much, so I will search also, and I will try to find the information after the
Yeah.
question. Okay, okay. Thanks, Abid. Yeah. What about yeah? Does it right?
Yazan, it's
Yeah.
Yazan.
Yeah. Yeah. Yes.
Uh, yes, I read about this endoditosis, which is done cell membrane to perform active transport. And
Yeah.
the second one, concentration gradient of particles like sodium.
Mm-hmm.
And the third one is the electrochemical
Mm-hmm.
gradient, like sodium, like
Mm-hmm.
potassium,
Mm-hmm.
potassium exchange,
Mm-hmm.
and there is the sodium.
Mm-hmm. So
Just
we are talking now examples, right?
yes,
Okay.
sodium.
Unfortunately, we can't hear your voice.
uh Sodium potassium bomb and exchange.
Yes, sodium potassium transporter.
Yes, and this
Yeah.
exotics.
Okay. Okay. So, what about your question? Do you have any questions?
For now, no. For now,
For now, no.
no.
Okay. 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 Okay, thank you, man. What about Solomon? Solomon, can you hear?
Yeah, I can hear you.
Did you do the task? Are you
Yeah,
ready?
yeah.
Okay, okay, man.
Okay. Basically, what I checked online, what I
Yeah,
saw based on my research is
yeah.
um the pumps are like they require an energy,
Okay,
which the energy input is in form of ATP.
yeah.
So there's a passive diffusion. Protein
Oh,
are specific to certain molecules, like the sodium potassium pump, like is one of the significant
uh-huh.
transport proteins.
Yeah, active
Yes,
transport. Yeah,
yeah, and based on the diagram here, they have like some kind of exchange
uh-huh,
that passes through like they use the ADP,
uh-huh,
ATP like for the transport.
uh-huh. Yeah,
And
yeah,
also um this what's it called? The potassium ions are
yeah,
also like they are almost like on the surface.
uh-huh. Yeah,
So uh I just have one question.
yeah. Okay.
Uh I don't know if it's possible like to break like to explain the diagram of the transport properly.
Okay, but
Maybe not right now, but maybe later. But I
yeah,
just want to get an understanding about it like very well.
yeah,
Though
yeah,
I can understand some things, but
yeah. So can you send this diagram?
yes, okay.
Okay, let's
I
send.
I don't know if I can share. Okay, 1 minute. How can I send from here? I don't know if it's possible.
What about to send to the chat? It's not.
There's no.
There's no doctor.
I
Uh,
can share my screen if it's possible. I don't know.
okay. So you are talking now about sodium potassium transporter diagram, or
Yeah. The active transport pump itself.
yeah, but we need the diagram. You have.
Okay, 1 second. I want to see if I can share it.
Mhm.
I don't think there's a way to share my screen from here.
Yeah, but you can actually put in the chat the link if you have the link,
Oh, okay.
and I will share. Okay, there we go. I will open it now. You see right now?
Yeah.
Yeah. So you want to explain which one? This one or Solomon? Which one you want? Not yet.
I can't see your screen though.
You can see my screen now?
No, no, no.
No?
It's just black.
Wow. Why?
Maybe you can tell him which picture, the number, second, first.
Go. What about you guys? Could you see my?
Yeah, I can. I can see the the stream. Yeah.
Yes, me too.
Uh,
I can see.
so Solomon, you can see, right?
No, no, no.
Uh, no. Okay, so maybe that's because of internet. I don't know.
Yeah, maybe.
Yeah. Okay. Then okay. Maybe later. I don't know how can. Maybe later or maybe next classes.
Okay.
Okay. Okay. Okay, guys. Thank you actually for participating in the activity because this is very important to my mind. You are studying now here to search yourself, to prioritize the information, make a list. This is very important to my mind. Okay, we just have left 15 minutes, so I will go quickly. We mentioned and spoke about all these organelles and part of the cell, so I'm not paying attention that much. Okay, now here just quickly, and very important information, mitosis and meiosis. So as you already guys mentioned, differences and Abed I guess mentioned or yeah. So they are the processes by which the cells reproduce itself. Most human cells reproduce asexually by mitosis, but the spermatozoa, spermatocytes and oocytes reproduce by meiosis. So germ cells reproduce by meiosis. Whereas the cells reproducing by mitosis finish up as exact copies of the parents. Okay, so what's the difference between mitosis and meiosis? So after mitosis, we will get two daughter cells which contain same amount of the chromosomes as the mother cell. You know that normally human cells have 23 pair chromosomes, so 46 chromosome. So after mitosis, uh, every daughter cell will have also same amount of chromosome 46 23 pair chromosomes. But when it comes to the meiosis, uh, by the help of the meiosis, amount of the chromosomes is divided, and only half of the chromosomes ended up in the daughter cells in spermatocytes and in oocytes. So in human case, spermatocytes and oocytes contain only 23 chromosomes, not pairs, just 23 chromosomes. Why is it important? What do you think? Why the human body and actually many organs, many living organism bodies needs these two processes, mitosis and meiosis? Why can't we live only with one? I mean, why was mitosis enough, and uh, why does meiosis exist and yeah, is developed also?
So we use sexual reproduction. We need like
Mhm.
both from like two sides, maybe the father and the mother.
Okay.
So yeah, by combining them, they make the first like cells,
Yeah.
which is like developed to a fetus in the future. So yeah, we need to actually.
So, but why can't the cells developed without meiosis just be developed by mitosis, divided, and then those cells which are produced by mitosis can also fuse to each other? Yeah. Uh, why?
I think we need like to like make differences and changes, so the randomity of like exchanging DNA between genes.
Yeah.
Uh, yeah, so that's why we need
Yeah.
to a father and a mother. Yeah.
Yeah, we need further. Yeah, yeah. That's very important aspect of meiosis because during meiosis, crossing over, so changing of the part of the maternal paternal chromosomes takes place. But also very important aspect that to maintain the amount of the chromosomes in the human body cells, that's a main reason why we need meiosis. Otherwise, without meiosis, after every fertilization, after every fusion, after every zygote formation, the number of the chromosomes will be multiplied, yeah, will be increased.
Yeah, exactly.
Yeah. So
So
yeah,
four and not two n, I
yeah,
think. Yeah.
and it will be increased. It will be, yeah. That's why meiosis happens to maintain the amount of the chromosomes in the cell. Okay, yeah, thank you, guys. Mitosis has a 4 stage: prophase, metaphase, anaphase, and telophase. What's happening in each stage? I'll just go quickly. So, during prophase, nucleus membrane is degrade, or how can we say? So, nucleus does not exist anymore. The membrane disappear. So chromosomes, so genes which are in the DNA, which are found inside of the nucleus, so ended up in the cytoplasm. This is the main aspect in prophase. And we have to remember, and you see here that by that time, by the prophase, each chromosome contains chromatids, so 2 daughter chromosomes. That means that those chromosomes is replicated during interphase. Yeah, during interphase has S phase, so replication phase. During S phase, chromosome, so DNA is replicated, and 2 daughter chromosome is produced. This is very important. So what's happening in metaphase? In metaphase, those chromosomes which contains 2 chromatids become attached to the spindle fibers. You see, they become attached to these spindle fibers. What about anaphase? During anaphase, the chromatids in each chromosome are separated. So as we already mentioned, this is very crucial, very important. Those chromatids, so two daughter chromatids, starts to separate each other, and they start to movement in each poles of the cell. So one chromatid from each chromosome then moves toward each pole of the spindle. You see here. And what's happening in telophase? In telophase, cleavage. So cell is cleavaged, and 2 daughter cells are produced. Each cells develop their own nucleus, and also organelles are divided, and we ended up, and we will have as a consequence 2 daughter cells which contains nucleus, and each nucleus, and they have also 46 chromosomes, because only chromatids was separated. So they ended up to have the 46 chromosomes, but each chromosome will not have any more 2 chromatids, just 1 chromatids. One, yeah. Guys, do you understand? Does it make sense? Do you have any question?
Yeah, I think I have a question. So I know
Okay.
after telophase, so the last one, the last phase, there's the term that we use to mention the end of the separation of the
Yeah.
So I don't know if you know that term. I forgot it. So that's my question, I think.
There was another term, yeah.
Yeah,
Prophase.
after telophase. After,
Yeah.
yeah.
Prophase, metaphase, anaphase, telophase. Yeah, I remember. Yeah, I know what you're talking about. Prophase, metaphase, anaphase, telophase. Is it written here? Uh,
It's the cytokinesis. Yeah, I do.
cytokinesis.
Yeah, yeah.
Cytokinesis. Yeah. Is it written here? Yes, cytokinesis. That means the cleavage of the cells, right? So,
Yeah, the end of the yeah.
yeah. Okay, yeah. Yeah. Okay, guys. Any other question? Okay, so we can continue. We mentioned cell cycle. We already know this cell cycle. So mitosis is just part of the cell cycle. Then there's G1 phase, where cell metabolically is active, duplication, so duplication of organelles, cytoplasm, cytosolic components, centrosome replication takes place. S phase, DNA is replicated. In G2 phase, cell growth continues, enzymes and other proteins are synthesized. And here we have G2, G0 phase for non-dividing cells. And now meiosis. Meiosis, which has a two-stage, first meiotic stage and second. So actually meiosis contains two division, and each stages have prophase, metaphase, anaphase, and telophase. So I will tell you quickly what's happening. So prophase 1 is similar to the prophase in mitosis, but however, instead of being scattered randomly, the chromosomes are arranged in the pairs. So we have 23 pairs chromosomes. Each chromosome contains two chromatids, but they now are not arranged in randomly. They make so each pairs are together, arranged in together. And we see in each pair of chromosome genetic material may be exchanged between two chromosomes, then the crossing over takes place. This is important for it's written here, it's these exchanges that are partly responsible for the differences between children of the same parents. Okay, crossover. So in metaphase, actually, same process takes place, but what's the difference? Here in meiosis 1, not pair chromosomes are separated. So if the in mitosis, chromatids was separated in each chromosome. Here in meiosis 1, just homologous chromosomes, so pair chromosomes are separated. Okay, so one chromosome from each pairs move to each. So they are now 23 chromosomes of each and each end of the spindle. And in telophase, cleavage of the cells takes place, and end of the meiosis 1, each cell contains 23 chromosomes. So 23 chromosomes, not 46 anymore, but each chromosome contains two daughter chromatids. And that's actually what's happening in second meiotic stage. In second meiotic stage, now in each chromosome, and we know that there's only 23 chromosomes left, each chromatids inside of the chromosomes is separated. And by the end of the telophase, we get two cells which contain just 23 chromosomes. Does it make sense, guys? Do you understand what's happening in meiosis? I know this can be a little bit difficult, but I think you should understand the concept. You should understand the thing. Guys?
It's not that hard, actually.
It's not hard, yeah. But you should understand. You should, yeah.
It's just a bit tricky. It's not that hard. It's a bit tricky. That's it. Yeah.
Yeah. And I don't know. It's already 7 o'clock here, so maybe I don't know. Guys, do you have just few minutes to finish? Do you have a few minutes to finish the lecture?
Yeah, for me, I have
Yes,
no problem.
of course.
You have another subject?
No.
No.
You have questions or?
No questions.
No question. Okay, so I will finish quickly because it's already 7 o'clock. So fusion of comments. It's okay. So organelles. We spoke about organelles. So we am not going to attention. Different tissues. We mentioned different tissues: epithelial tissue, connective tissue, muscle, nervous tissue. And in your book, each one is quite well. There's epithelial tissue, and the epithelial tissue is classified in two ways: by number of the cell layers, for example, simple, stratified, shape, squamous, cuboidal, columnar, and you see the examples. I'm just go quickly because you will read this information or listen from my lectures, which I already record. So glandular epithelium, connective tissue, bone. And so examples of the connective tissue is the bone, cartilage. Dense connective tissue is the adipose tissue. So loose connective tissue is the areolar tissue, adipose tissue, reticular tissue, blood. And example of the muscle tissue. An example of the muscle tissue are the skeletal muscle, cardiac muscle, and smooth muscle. Yeah, I know that you know that I'm cardiologist, so cardiac muscle is I don't know. I can speak a lot of about it, but I guess I will speak and we will discuss this topic later in cardiac chapter. But you have to read books and also listen my lecture. Nervous tissue and neuron, which is the main cell, has a peristalsis. Again, tissue repair. Several sentences is mentioned for tissue repair. Okay, guys. So I think that we mentioned main topics. We just don't talk that much in tissue, but it's okay. In next classes we mention all of them because we will speak about them. Okay, here is at the end of the this chapter, you see the different activities. First of all, there are the still questions to test your knowledge, and I encourage you guys to answer the question. First of all, do you have access to the book? Is it uploaded?
Yeah, I don't have it on my drive. I don't have anything actually this semester.
So it's not uploaded.
No, it's not.
I send it to the Salome, your coordinator, and I will ask her. And I have it, and also you can. I can also send it to you, but this is my mail. I will write down my mail, guys, in our chat. And in this case, that Salome will not provide it, I will. Or you will don't find. I will send to you. So, guys, just mail me. Okay, just several slides and I'm finished. So please answer these questions by yourself. They are different activities, so fill in the blanks. If you will have time and if you want to do some exercises, this is a really good one. Link the blanks and here are the words. Label the diagram. This is about plasma membrane. Word search. You can do this crossword even here is can do this, and at the end of this chapter, and every chapter is a glossary. You may don't know any terms and you can search these terms. Okay, guys, this is actually what I wanted to speak and say in very first tutorial. So what's our plan? You will have online classes like this next week. And I will record the lecture also next week. Next week's topic will be cancer. So I will record a lecture about cancer and we will discuss the cancer together. But I will ask you also some questions about very first this chapter which we was discussing today. So please read at home, be prepared. and I will ask you some questions. No, I think you already know most of the things. Okay, that's all I wanted to say actually. Do you have any question? Or otherwise we can finish our online class.
About the lecture, is it live or is it just recorded? So can we join the lecture?
Lecture is I record it for the first week, but I guess it's up to us. What kind of lecture do you prefer? You prefer online lecture? We can change this rule. And
I mean, can we do it live and still have the record, you know?
uh, uh, yeah, actually. Actually, what we did now, it was the you mean that let's have the lecture and live and record this and upload this, yeah?
Yes,
Like
yes,
this.
exactly. Yeah.
Yeah, we can do it actually. Um, yeah, we can do it somehow. But I don't know exactly actually. I will ask Salome. And there are several options we can make. Yeah. So yeah, I got it. Yeah. Then we now I will speak to Salome about it.
Okay. And about the drive and the books, we just send you an email, I think.
Yeah.
Okay.
Duru Mamrikishvili. And I will ask Salome. And because I already sent to Salome this book, PDF version of this book, and I told to her that, and she told me that she will upload, and I will just tell. And if you if you don't find, if you will not find book on Drive, mail me. This is my mail. And yeah.
Okay, thank you.
Thank you, guys. Thank you. It was really interesting for me. I really like the uh, what I'm doing in this profession, teaching. So thank you for listening me. Thank you for actively involving this lecture. And see you. Till next time.
Yeah, thank you. Have a good day. Have a nice day.
You too. You too,
Yeah.
guys.
Okay, bye-bye.
Bye bye. Okay, so how can I? Okay, yes. Stop record.